There is a lot of changes in the genome of CLL, and this is known since many years, but the epigenome, which is actually the science that studies the gene regulation and expression of genes without changes in the DNA sequence, is much less studied. So I saw that as a window of opportunity several years ago, and when I started the research group on epigenomics, we focused on CLL because it was not very well characterized from that perspective. And we identified many interesting things. One of them by studying the DNA methylation profile of the disease and compare the DNA methylation of the disease to normal B cell differentiation. We observed actually that we could identify 3 groups of patients that seemed to be derived from B cells at different maturation states. We called one of them naive-like, more similar to naive cells, another one memory-like, more similar to memory cells, and another one that we didn't know what that was, the cell of origin, but we call it intermediate because it was in between the two. When we correlated that with the clinical data, we saw that these 3 groups have strikingly different clinical behaviors. And that was, I think that was a very interesting finding, because there was a study on the pathophysiology of the disease that had also an impact on the clinical behavior of the patients, which is something very nice to see.